this session, I'm going to focus on some of the work I did for my PhD, so mainly looking at the canola effects on nitrogen and soil nitrogen. But first, I wanted to reiterate some points that have already been made at this conference, and basically justifying introducing canola in the rotation. And so you've heard that as much as a 20% wheat yield bump may occur after growing canola. This research has been performed in Australia. They've pioneered this yield bump idea where it's having canola in the rotation can have a rotational benefit. So not just where you can try to tease out the nitrogen effect versus the rotational effects. And they've seen that canola can actually bump yields through the depression of suppression of diseases. And so here's a graph right here on the right-hand side, which shows the yield or wheat yield here, either following wheat or canola on the y-axis, and nitrogen additions on the x-axis. And what you see here is, so the empty dots right here, these are the observations under no disease pressure, where the black dots here are the yields that were recorded with incidence of take-all. And so you can see here the important thing to show is that when you do have disease suppression, it limits that nitrogen response, or that response of wheat to the nitrogen additions. And the other thing to notice is that in all cases, except for this one here at the highest level, wheat following canola had greater yields than wheat on wheat on wheat. And so this is somewhat attributed to this rotational effect of disease suppression. And so it's just important to keep in mind that part of the big picture and the rotational aspects to keep in mind, not just nitrogen. However, other research, so first you can have healthier roots when following, or healthier wheat roots when following canola. But then also the Australian researchers have shown that more soil water and nitrogen is extracted from soil when you have canola wheat versus wheat on wheat. And so it may not just be due to healthier roots, but also looking at the root architecture or the root structure. And so Ashley Hammock from our lab performed a study a few years ago which showed that canola actually has a different root structure than, let's say, wheat. So we've heard about that in this session, about how wheat has the fibrous roots, canola has a big taproot that Taylor will talk more about. But also looking at the root hair geometry. So you can see here, the dots in blue, this is canola roots. And so you have more, and it's pictured right here, you have more longer root hairs coming out from that root. And so you're able to extract more water, more nitrogen. And then I just wanted to also mention too, you've probably heard about this, but introducing canola in your rotation is going to shift your nutrient management. And so we know here that canola versus our white or our dark northern spring wheat, canola has a higher nitrogen requirement. So basically more nitrogen is needed to produce a given amount of grain than wheat. And a lot of that has to do with the canola is more dense in nitrogen and carbon, so you just don't produce as much volume or weight of grain as you do canola. But this is not due to a lower uptake efficiency. So canola is actually pretty good at scavenging the soil of nitrogen. So it's not a lack of pulling it up from the ground. It's more to do with how the plant utilizes nitrogen. And then this is something that Bill Pan talked about today, about how we have to think about how these different alternative crops to wheat utilize or demand nutrients in the soil. And so this is just to show here that Bill Pan pointed out that spring canola responds to boron. And so to keep that in mind too, of when you are going to grow canola, to think about different nutrients that may be deficient. And also to nutrient placement as well. And so this is work that was done in our lab, which shows how the taproot that comes down from canola, it can actually exhibit ammonia toxicity if it runs into that band. And then that can affect the nutrient or the root development after that. And it's not like wheat where it has that fibrous structure. And so that's also important too. But what I really came here to talk about, though, is the nitrogen dynamics following canola. So this is the meat of my research. And basically what I looked at is, do we see an increase in available nitrogen after canola versus field pea and wheat? Okay, and so first I'm just going to show what other people have shown or found in other places. So this is a French group of researchers. And so is everyone familiar with net mineralization, net immobilization as a concept? And so basically net mineralization is you have a release of nitrogen from soil organic matter. And net immobilization is a tie-up of nitrogen from the soil. So you have a depression in the amount of nitrogen that's available to the plants. And so here in this study, they added alfalfa shoots to soil and saw here that there was a release or more nitrogen that was in the soil versus a soil that was unamended with any plant materials. And then they added mature canola leaves. So this is after the leaves have senesced or dropped from the plant. And you can see here is that when it was added, there was initial suppression in nitrogen in the soil, so there was less available for a period of time. But then the nitrogen started to turn over, and at this point here, you had more nitrogen available than that soil that had been unamended. But in another set, so they took another batch of mature canola leaves, and they found the same depression in soil nitrogen, but that nitrogen never quite turned over. And so throughout this 169-day study, there was less nitrogen in that soil after this batch of canola leaves were added compared to the soil that was unamended. So think of that as like your fallow period. So you don't have a period of leaves decomposing and a demand for the nitrogen. And then here is maize straw or corn straw, where you have a depression of soil nitrogen in the soil. But here, you don't even have some turnover. It's just flat lines at that bottom. And so these researchers tried to find out a characteristic of that plant material that could help predict this dynamic. And so C to N, who's familiar with C to N ratio or carbon to nitrogen ratio? So it's a measurement. So if you have a plant residue, you can measure the carbon in that plant residue or chaff or straw, and then you can measure the nitrogen. And then you look at that ratio of the two, and that gives you the C to N ratio. And it's an indicator for its ability to, how quickly it decomposes, and it allows you to predict the availability of the nitrogen. And so you can see that the C to N ratio, the plant size C to N ratio that was less than 24:1, so that has a lot of nitrogen associated with that carbon. Those were the only plant residues in the study which had a net release of nitrogen in the soil, whereas C to N ratio greater than 24:1 had a net tie-up. Does anyone know what crop residues with a C to N ratio crop residues may be? So it's quite high. So my residues have ranged between 60:1 for field pea and up to 150 for wheat straw. And so that's why we hear a lot about nitrogen tie-up. That's why on your fertilizer sheets where you determine the amount of fertilizer that you need, you have that debit. So you debit the nitrogen that would be tied up if you're producing a crop after winter wheat. And so in other studies that have looked at the effect of canola on soil nitrogen, there's a big range in the literature, so there's a discrepancy. Some researchers have said, there's more nitrogen available after you produce canola versus wheat or field pea, while others say there's less nitrogen. Canola behaves more like wheat, where you have a greater amount of nitrogen tied up, or you have nitrogen immobilization. And so a lot of that can be due to the quality of that residue. So canola has a wide range of C to N ratios. It depends on your nitrogen fertility. It depends on the amount of nitrogen, residual nitrogen in the soil, so not just your fertilizer additions, as well as other factors too. And so that can influence the availability, and that's why you may see discrepancies in various studies done. And then also too, the idea of nitrogen sequestration. We talk about carbon a lot, but as you saw in the panel earlier, nitrogen sequestration can be important because you're trying to build up that soil organic matter. If you remember what Dave Huggins showed with the Cook Farm and showing the increase in total soil organic nitrogen with time, that's important because this is a source to slowly turn over nitrogen with time. And you can see here that canola, it releases some of its nitrogen, so it looked like it released 40% of that residue nitrogen, and then it stabilized. And this was also shown with field pea, whereas wheat, it lost about 20% of its nitrogen and then started pulling in or tying in nitrogen. And so that balance between release and stabilization is important, and it influences the availability of nitrogen after that crop into the next season. And it affects your nutrient management as well. Okay, so this data is from my PhD. And so what I wanted to do was look at these different residues. So these were mostly taken from the field. And I wanted to look at the amount of nitrogen in these crop residues that had a range of carbon to nitrogen ratios. And I also wanted to look at where that nitrogen was in the plant. So what sort of carbohydrate fractions it was associated with. And then also relate that to the nitrogen dynamics when you added that residue to soil. And what I saw was most of the nitrogen in crop residues, the majority of that was associated with soluble components. That means that if these residues were to interact with water, basically that nitrogen would leach out of the crop residue and then be available to interact in the soil. And this was especially true for pea and canola, and less true for wheat. And then the other thing too is that so canola, wheat, and pea along here, those are all crop residues collected from the field at crop maturity. And so you can see here that the residue or the C to N ratios are quite high. And if you remember the first graph that I showed, it was 24:1 was the break point between that net release nitrogen and net tie up. And so we may expect these to tie up nitrogen when added. Now this was a field pea that was heavily fertilized. And so you can see that it has a really low C to N ratio. And you would expect it to behave more like a green manure. And so if you're cover cropping, that's where you would want a residue that had a low C to N ratio. And so we added these to soil. And so this was the residue that I just discussed, where this is more like a green manure, field pea. And you can see here that when we added it to soil, we had a slight depression or a slight tie-up of nitrogen, where the microbes started to react with this residue, consume some of the nitrogen, and then there was a release. And so we have a net release of nitrogen. So that means there's more nitrogen available in that soil when this field pea was added than the soil without any residue added. And then we added the other residues, and we can see that the residues that were grouped with around 50 carbon to every 1 nitrogen. So it's a moderate C to N ratio. There was a net tie-up of nitrogen. And this occurred whether it was canola, pea, or wheat. And it's interesting because we usually associate nitrogen tie-up with wheat straw and not necessarily pea and wheat. Pea and canola. And then we had another group of residues that are shown here in red that also had a depression in soil nitrogen availability, but even more so. And so these were the residues that had really high carbon to nitrogen ratios. And so those are really poor quality. Those are going to tie up the nitrogen. And so when I tried to predict the nitrogen release, I found that the amount of nitrogen that was immobilized or tied up in this system, that correlated with the amount of nitrogen in the plant residue. And it didn't matter what type of nitrogen that was, a total soluble nitrogen, the amount that dissolves readily in water without any sort of treatment or digestion, and then water extractable nitrogen too. But I found that it didn't correlate really well with the carbon to nitrogen ratio. And that's because there seems to be a break in this relationship. So right here, you can see that it has a pretty good relationship at this low C to N ratio with saying, okay, if you increase the C to N ratio this much, you would expect so much nitrogen to be tied up. But then this relationship breaks right here. And you can see here that as you get more and more and more carbon, less and less nitrogen, that a lot of nitrogen's tied up despite those large differences in C to N ratio. And this is important too because I mentioned that the crop residues tend to have C to N ratios from 60 to 150 to 1. And so that means that we would expect that pea, wheat, or canola with these residues to have or to tie up nitrogen. And so this kind of challenges our thinking a little bit because in our fertilizer sheets or trying to figure out how much fertilizer to add, we often credit P, right? You're familiar with you credit P about 20 pounds of nitrogen per ton harvested, and you debit wheat. And so this challenged my thinking as well. And a lot of this has to do with the interaction between quantity and quality and the background, what's going on in the soil too. And so basically it's all relative. And so in our systems, P ties up a lot less nitrogen relative to P for two reasons. And so one, P generally has more nitrogen than wheat. It generally has a lower C to N ratio. And the quantity, we produce a lot more wheat straw than we produce pea straw. And so this graph right here was taken from 3 groups of Canadian researchers. And so their wheat, they only grow spring wheat, and their straw yields are a lot lower. But you can see here that this interaction is important because in this first case here, you see that canola tied up the most, or you would predict that canola, based on its characteristics, would tie up more nitrogen than pea and wheat. But in the next set, it was actually pea, and then the final set, it was wheat, and then wheat again. And so this explains why in some studies you can see that canola behaves more like pea or it behaves more like wheat. And it has to do with that relative effect. And so in our systems where you have a lot of wheat straw that have poor quality, that's why we end up debiting. And then in the systems with pea, where you have more residual nitrogen after cropping, as well as a lot less immobilization or nitrogen tie-up, we end up giving that a credit, relatively speaking. And so in conclusion, all residue types resulted in initial nitrogen immobilization or tie-up. The extent of immobilization was related to nitrogen partitioning into fiber fractions, specifically that soluble fraction, the one that can dissolve and react readily. And then there was an interaction between quantity and quality of the nitrogen fractions in the crop residue, and that's affecting nitrogen cycling. And are we going to do questions or do panel? Okay. Okay. So we're talking about 2 crops, an intermediate rainfall zone, a canola that yielded 3,000, and then 90 bushel winter wheat.
And then the stubble's just left standing.
Okay. So what I expect at harvest is that you would have a drawdown of nitrogen in both profiles. So to what Bill Pan presented today, canola is really effective at getting down to the lower depths, just like wheat. Has a high uptake efficiency, so it can extract a lot of nitrogen. But what I've seen in the higher rainfall zone is that you can lose. I found that the leaf nitrogen for canola is pretty important. So you can, in my studies, I lost between 25 and 50 pounds of nitrogen per acre in just that leaf drop alone. And that seems to turn over faster. And so I saw an increase in apparent nitrogen mineralization compared or following the canola. But I didn't observe that in the intermediate zone. So in Davenport, Washington, I didn't see an increase in mineralization. And that could be for two reasons. There was a lot less leaf nitrogen that was dropped. And then just the lower rainfall maybe has a slower rate of turnover. And so I think it depends on the year, the amount of rainfall that you're getting, and how quickly that leaf nitrogen. I think the leaf nitrogen is an important component of the canola, more so than the pods and the chaff. So part of my research too was looking at the timing of mineralization, so the turnover of the soil organic nitrogen, and I saw that in the higher rainfall zones, about a half of that nitrogen turned over before April. And then in the intermediate rainfall zone, it was less. It was about a third of that turned over before April. That was after canola.
So my question is, would you expect to see a difference between the wheat and canola?
I think
7 months after they were harvested, because they started out the same when they were.
So I don't have data on this, but I would hypothesize that if you do have enough leaf drop that contains enough nitrogen, then yes, you would see more residual nitrogen in April after canola than wheat. But I think it depends on that pool of nitrogen. Yes. So the question is, should you take an in-season leaf test?
Well, I don't know, when would I take that leaf test to kind of get an idea how much nitrogen the canola would, that's really soluble, because you said it depends on that carbon nitrogen ratio.
Okay, yeah. And so you want to know about a leaf test for the subsequent season to give it a credit. Okay. Well, part of this work that we're trying to do is to answer the question. So if you look at extension bulletins, you have a credit for P, and that's when I mentioned it has higher nitrogen and lower biomass that you're returning, so it has a lot less effect on the nitrogen availability. And wheat, you debit. And so our thought was, well, do you debit after canola or do you credit? And where I stand right now, my hypothesis, and I don't quite know the answer, but what I'm deducing is you would give it neither a credit or a debit. So I'm thinking that it falls in between wheat and it falls in between wheat and pea in this region. And as far as a tissue test, I mean, that's part of the work that we're trying to do so that you don't have to do that to get an idea. But I mean, that goes back to an important point though. So with canola, another reason why the nitrogen use efficiency is so low is because it's less efficient than wheat of reallocating that nitrogen to the grain. And so that's why you end up with a lot of nitrogen in the vegetative tissue. And so you have these lost pathways like the leaves, and then you have nitrogen that's remaining. I think the nitrogen harvest index is pretty low. And so there is that pool of nitrogen to recycle. But I don't know, I'm not quite sure on how to link that to a tissue test and link that with a management decision in the next season. Bill, do you have any ideas?
Well, I guess this is
So Bill just mentioned that, I'm repeating this for the cameras in the back, but so Bill mentioned that there is a pool of nitrogen that's lost mid-season that could behave like a green manure and influence the availability, but we're not too sure the timing of that. But I will bring up one study that I know of with winter canola that was performed in France. And this one group of researchers were finding that up to almost 100 pounds of nitrogen per acre was lost from the winter rosette to the spring rosette. And so they labeled the leaf with a tracer so they could account for how much of that was recaptured in the spring by the plants. And they found that 30% of that nitrogen was recaptured in spring by the plant. So it turned over winter and under sufficient moisture conditions and before the temperatures got too cold to inhibit the microbes. But 40% of that was lost to volatilization, and that's because it's just sitting on the surface. And so it's able to interact with the atmosphere. So, but it's a good story and a bad story at the same time, you know. So you do have losses, but you do have recapturing, which goes back to your question too, with in April, which is I think related. That was in the Mediterranean climate, higher rainfall, like Pullman. So that's where that would influence.
Okay, so I'm going to talk about a little bit of biology today, and I'm just going to try to not bore you too much with lipid structures and cell membranes, but just to explain, what I'm looking at is the cell wall of microorganisms that reside in the soil. And the reason for this is in a survey conducted by the oilseed group, one of the areas of interest were the rotational effects of canola on soil quality and soil health. There's been some work done on this, and I am no means an expert on this, but I have found some pretty cool preliminary data that indicates that there are some differences in the soil community. So that's the first thing you have to do is, right, you have to prove there's a difference before you proceed. If there's no difference, then we just go on to something else. Our experimental data is generated from plots that are in Davenport. They're on Hal Johnson's land. It's a complete randomized block with 6 reps of winter wheat, Finch variety, and 6 reps of canola. And those, the typical rotation for Hal Johnson is winter wheat, spring cereal, and then a fallow. And the setup for our sampling is that we sample in the spring after the winter wheat and the winter canola have had a chance to establish. Microbial communities are very active in the springtime. There's plenty of moisture and it's not too hot yet. And then the sampling that took place would be, let's say for the 2011 plots, we would have sampled in crop in the 2011 plots in the spring. And then the 2012 and 2013 samples represent samples where we went back, sampled from the plot that we originally sampled from, and it was under whatever crop was next in the rotation. Normally coming out of the canola winter wheat plot, it was going into spring wheat. So the first year, normally we were sampling in spring wheat, and then after that it was either fallow or another silver grain. The analysis that we're conducting, and I'm not going to talk about all of these, I'm going to focus on the first one, which is PLFA, phospholipid fatty acid analysis. But we are going to be looking at other nutrients like carbon to nitrogen ratios. We're going to be looking at other enzyme activities just to give us an idea of soil health. So what is PLFA? PLFA is what I call the soil microbial census. We're very familiar with the census and the census data that is generated for the United States, and each state and each community have these data. And so when I think about microbial communities under different cropping systems, I think about 2 different communities in the state of Washington, one being Pullman and the other one being Seattle. Obviously, there are differences in those communities. Our climates are fairly, temperate, a little bit different. They get more rainfall, we get less rainfall. But what is it that generates those differences in the communities? You can see that, I'll focus on a couple of these. I thought it was interesting. They have a lot more people. They have a higher crime rate. If you want to buy a house in Seattle, you're going to pay twice as much than you are in Pullman, although $219,000 for a house is a lot of money, in my opinion. So So there are differences in ethnicity, there's differences in the way people spend money, there are differences in education. Pullman has a higher high school diploma rate and a higher bachelor degree rate than Seattle, so go Eastern Washington. So this kind of sets us up to talk about how differences in microbial communities exist. And we know that they're always changing and what causes those changes. The way we can analyze that is through the lipids, extracting the lipids in the cell wall. So just really briefly, I will point at the phospholipid bilayer. The cell wall is made up of a bunch of fatty acid lipids. We know, we're familiar with lipids because we're here, because we're growing canola. The lipids are what are extracted from the canola, it's the oil. And those lipids are contained in the cell wall. Those lipids will be unique to a different species, whether that's a plant or whether that's a microbe. And so we can get some identification information out of the nature of the lipid, but I also will say that sometimes the nature of the lipids change as a plant response to stress, whether it be drought or whether that be cold tolerance. And so we can kind of monitor the health and the diversity through these extractions. And so I'm going to emphasize preliminary results here because these are fairly rough, crude statistical methods that I've analyzed the last few years of data, with a lot more to come. But what I wanted to show you is starting with the 2011 plots, and then also sampled in the spring of 2011. So we're sampling in crop. What I've got here are 3 different depths. The yellow and black represent the canola, the crimson and gray represent the winter wheat, and the bars indicate the standard deviation or the error, or how much the data are spread out. So you see a lot of overlap in the bottom 2 depths here, and at the surface depths, you see not a lot of overlap, so we can deduct that those soil microbial communities at the surface are different. And just focusing in on those 2, the circles represent the area that would be occupied by the microbial communities statistically with lipid analysis. And so there is some overlap, there's some commonality between the 2, but there is a majority of the lipids are different. And so once again showing that there are differences in microbial communities. Now we go to the year after the crop, so this is in 2012, in spring wheat, and those error bars get a lot bigger, a lot longer, and we see a lot less separation. There's more commonality. Focusing in, those, as you can see, they occupy a lot of the same area statistically, so we wouldn't be able to say that these microbial communities are different. So I was scratching my head. I thought, okay, we've got differences in year 1. Those quickly diminish in year 2. Going to the 3rd year, similar results. There is a little bit of separation with the lower depths, but for the most part, our 2 upper depths is fairly congruent and very similar. Advancing to the 2012 plots, the story got a little bit different here. Saw a lot more separation in current crop as well as subsequent years. And so you see that those aren't laying right on top of each other. They are definitely different. And looking at those on the depth, individual depths, we see the 2 circles where they overlap, there's not a lot of common area there. So at all 3 depths, now we've got microbial communities that are very different. Looking at the year following this of the 2013 plots in spring wheat, still a fair amount of separation. Some of that separation is diminishing. However, there is still a fair amount of separation in the microbial communities. The surface and 5 to 10 centimeter depth show a lot of separation. The lowest depth doesn't necessarily show a lot of separation. That's just because of the variability in the microbes under the winter wheat. There's a large error on the PC2 axis. So my conclusions from preliminary results are that, yes, there are differences. if you can imagine how much data that we're generating, so we all know that soils are full of microorganisms. We take 2 grams of soil and we're extracting the lipids out of every microorganism in that soil. So when we run our samples on the gas chromatography, then we end up with lines and lines and lines and lines of data. So that's why I said this is a crude, rough estimate of what the microbial communities are. So my concluding statements are that based on preliminary results, there are differences in the crop, particularly in the current crop. Those differences may subside with subsequent years. However, we saw that some of those differences existed in the second and third year. I'm not going to hypothesize yet as to what those differences are, what is causing those differences. TIE's data may explain some of this, but our future work now is to move on to future work. Our future work now is to take additional samples and then dig deeper into the data of the PLFA. Run these through a lot of multivariate analysis so we can start to tease out what the differences are. So we're going to start looking at communities, bacteria, fungi. What's the bacteria to fungal ratio? We're going to look at what the microbial biomass is. We're going to look at the health of the community, differences in saturated versus unsaturated fatty acids. That will be an indicator of stress. So if we see an indicator of stress following canola, then we can say that there may be something there stressing this. If we don't see that shift, we don't see a stress indicator, that's good. So essentially, we're just going to look deeper into the data, and we're going to try to decide what the differences are caused from and what those differences mean. Are those differences good or bad? The hypothesis now is that those differences are not bad, but if we can identify what the differences are, we can manipulate our systems a little bit more to increase our production. I'd like to acknowledge the help of John Jacobson, who is working out there in the plots, Tim Smith, who previously worked out in the plots, and Hal Johnson for providing the land of which the research plots sit on, Bill Schillinger for serving as my major professor, and Ann Kennedy for the use of our facilities to conduct this research. With that, I'll entertain. We didn't see an overall increase, but keep in mind that what we're looking at are we're looking at log numbers. So the differences would be between 1 and 100. And so the difference between 1 million and 2 million aren't necessarily detected. So we didn't see a lot of differences overall, and that's attributed to most of the microbial community is going to be the same whether you're in winter wheat or whether you're in winter canola. It's like the Seattle Pullman analogy. There's 76% white Caucasian and 78% white Caucasian. So the base or the majority of those microbes are going to be the same whether, depending on whatever crop you're in. However, it's the other 25% of the community that is more susceptible to environmental effects or maybe crop residue effects. So no, I didn't see a difference in population numbers, but now we just need to identify the differences in the makeup of the community and where those take place. To repeat the question, we have another study that is looking at the camelina in rotation, and that's at the Lind field station. Those samples are currently actually being run on the GC as we speak. That data is soon to come after I finish my soil chemistry homework.
All right, so like Jim said, I'm going to be talking a little bit about my thesis research. And basically it focused on comparing wheat and canola residue and how their different compositions may affect soil crusting and to a certain extent subsoil compaction. So why are we concerned with soil crusting? Well, in arid and semi-arid areas, it can really cause issues. It can reduce your water infiltration, water runoff and erosion, and then most importantly affects seedling emergence, as you can see in this picture down in the lower left. So to better display this, I did a scanner experiment in our lab, which we fill a trash can up full of soil, and then we plant seedlings next to a scanner, and then it can take different pictures at different times. And so these are just some videos I made to show the effects crusting can have on seed emergence. Now these seeds were planted about 1.5 inches down, and then I would just like to note how small the actual crust is at the top that we got. It's probably only a few millimeters thick. This is the canola seedling. And then it just stops right there. And then the wheat. So it can cause a lot of issues with emergence. So crusts are formed mostly after a rainfall event. So the rain comes down, hits the soil, and that breaks apart all the smaller pieces. And then they get redistributed into all the nooks and crannies in the soil. And when the sun comes out, it dries that and it creates a small film on the top of the surface. And that causes, reduces water to be able to infiltrate through the soil. But in ag systems, when you add crops in there, you have all these other types of effects. And it can be affected by soil type, how much sand, silt, and clay you have in your soil, percent moisture, pH, organic matter, and then what crops are you growing. So my research was mostly looking at these residue effects and with a primary focus on silica. Because once these residues break down and the silica gets released, it can absorb onto soil particles, causing the pore radius to shrink even smaller, which can contribute to a thicker crust, or more potential for crusting to happen. So in the late 80s, Brown and Mahler did a few studies that had linked these higher amounts of silica to soil pan formation down in the lower profiles. And I think Bill showed one of these pictures earlier in one of his talks. But you can see here on the right, it's only about 3 inches down, and then you hit that first pan. So it's not a lot of room for the roots to be growing in. And then over on the left, it's even more distinct with the coloring. You can see right where that pan line is. So that's why we're kind of taking this idea, if silica is being linked to pan formation in lower profile, is that same thing happening on the surface? And then do these crops affect it? So why do different crops accumulate different amounts of silica? And that's mostly due to the root systems. So grass crops such as wheat tend to accumulate more versus a canola crop. And so when it gets absorbed up by the roots, it then gets distributed through the plant. And then when you harvest, it breaks down the soil. So in order to look at this, I did multiple studies, but today I'm just going to talk about a greenhouse study and a soil incubation. And so we grew soft white spring wheat and spring canola in a greenhouse under 4 different nitrogen rates. And then in the soil incubation, I collected soils from 2 fields. It was a Ritzville silt loam. One field had been cropped in cereals for over 50 years, no canola in the rotation. And then the other field had canola present for approximately 25 years. And so with these soils, I added silica amounts to that just to see what was going to happen. So part of the greenhouse results, this is the fiber analysis, and I just want to point out that wheat accumulates more hemicellulose than canola and similar levels of cellulose, while canola has higher levels of soluble material and lignin. So now to what I was interested in was looking at the silica amounts. And as you can see, the dark blue bars are the wheat and the light blue is the canola. And keep in mind that this is grams per plant, so it's on a very small scale. But if you think of this on a field size, when you got tons of residue, then it really becomes larger numbers. So wheat accumulates at least 4 times as much more silica than canola. And this residue is everything combined, so it's leaves, stems, pretty much everything except for the grain or the roots. So taking that knowledge, we applied that to the incubation, and I applied these different rates of silica down at the bottom. And this is amorphous silica on the y-axis here, and that's basically the exchangeable silica available in the soil that's going to absorb to the soil particles. And in the wheat rotation, starting out, it had way more silica available, so that's why you see that difference in between the 2 rotations. And then, of course, additions increased the amounts in the soil. So crust thickness, I would just measure this using a little caliper, but it didn't really show too many differences with increasing levels of silica or between rotations statistically, but physically you were able to see a big difference while I was conducting the experiment. And then surface resistance, this is measured just with the little penetrometer. And again, the wheat rotation was significantly higher for each addition of silica, and then it increased with the silica additions as well. So knowing this, we saw all these patterns. I was like, okay, this happens in the lab under perfect conditions. Well, what about the real world? Is this really happening out in the field? And so I went out, I just like to say, I went out one day to one field, or multiple fields, but this was only like a snapshot of what was happening in the field. So it was one data point. And I used a penetrometer, which I don't know if anyone's familiar with it, but it looks like a pogo stick. And then you push it down and it measures the strength it takes to pass through per inch. And so that's what this graph is showing here. On the y-axis, you're going down in inches, and then on the x-axis is strength. And so I measured 3 different fields. The blue is the cereals rotation with winter wheat, barley, and spring wheat. The green is winter wheat with canola and barley, and then the red is canola, winter wheat, barley. So as you can see, the ones that didn't have canola that year were much stronger, had much higher strength, going down to about 4 inches. And even in the cereals rotation, I couldn't get the penetrometer to go past 4 inches. And then the green bar, which has canola in the rotation a year before that, I was able to go a little bit further, but still hit that layer. But the canola, I was able to go down all the way to 12 inches, and I probably could have gone down farther, but that's just the length of the tool that I had. So I also took soil samples at these different depths, 0 to 4, 4 to 8, and 8 to 12 inches. And at least in the top, they followed the same pattern. So the cereals rotation had the highest amount of silica, followed by the green bar and then the red bar. But then down at these lower depths, it kind of gets, there's not really much of a pattern, except for in the canola rotation. It's pretty much the same all the way down. And so the theory is that the roots are breaking up these pans here at this, about the 4 inch, and then farther down at like 6 inches, which is causing water to infiltrate more and nutrients is able to move farther down through the soil profile. Bill also showed this in his talk earlier today, but this is just a picture of some roots that we had found that have the J hook right about 3.25 inches, which matches up pretty perfectly to this data here at this 4 inch mark. So even the canola taproots are having a hard time breaking through that first pan. And they're only growing out in about 3 to 4 inches of soil. So at least this data confirmed what I had found in the lab incubation, that wheat contributes more silica to the system, and then that leads to higher surface crusting potential and soil compaction. And then the canola roots seem to be breaking through those pans, aiding in nutrient cycling. Questions? Yeah, I'm not sure exactly why that is, but I do know that, I mean, that pan is pretty intense. I tried to bust through that thing, and it was really hard. So I'm guessing that the first canola crop maybe is starting to kind of break through that pan, but just doesn't quite have enough oomph to it. And then the second crop will come in and is able to bust through because that first couple inches might have been affected. But the question was, does silica contribute to the mellowness of the ground? And I'm not sure. I don't think so, but it just contributes to how likely it is to compact or to kind of stick together, but I don't think that's affecting what you're talking about. Okay, so in the lab I used it's called a pocket penetrometer. It's basically just a mini penetrometer, and it looks like a pen that you just push down, and then it measures how much it takes. I had to get a few different ones, so there's some that are used for like fruit ripening testing, and then there's a big range of variable ones that you can get that have different strength testing. Yeah, and he mentioned that the canola, after he harvests canola, that it absorbs the water much faster than the wheat soil does, and that I actually went out to those fields and tested that, and it's true. I saw that as well. So the question was, have Ty and I talked about the different biochemical properties and the different decomposition rates between the two crops? And the answer is yes, we have talked a lot about that. And we actually did, I have those slides with me, a different study. Yeah, I knew that question was probably going to come. So we took, as I mentioned in the greenhouse, we grew them under different fertilizer rates. And so we looked at these different rates that were grown both in the greenhouse and in field settings. And so this shows the decomposition rates. And you can see canola with no fertilizer, canola that was grown in the greenhouse, and then the wheat the same. And so basically we didn't really see too many differences with how they decomposed till about week 8. And then they kind of started to spread out a little bit. And then this is just showing how the silica itself was being broken down. So it had kind of a steep incline at first, and then it started to plateau right about that same point at week 8. So it seems like all of the residues, no matter what their different compositions are, kind of have that same window through week 8, and then they kind of spread out a little bit.
Yes, so the question is if would we be able to detect the amount of mycorrhizal populations associated with canola? Yes, mycorrhizal hyphae have a distinct PLFA marker, biomarker, and if there are diminishing or increasing amounts of that particular fatty acid or that makeup of the fatty acid, we should be able to detect that. However, if it's small, it might fall below detection limits. I'm currently requesting a mass spec detector so we can maybe increase our detection limits. But if there are big shifts or if the population is sufficient to detect, we should be able to track that.
So the question was, is there a difference in varieties and how they accumulate nitrogen? I'm not exactly sure how to answer that question.
Well, Tammy Stubbs did work on that, and Tammy worked with Jeremy for years. And
yeah, there are differences between market classes for sure, but even within market classes, say winter wheat, there are differences between varieties, and her work was trying to match up decomposition rates with rainfall zones, so that in the dry regions you could have a fast decomposing residue. And I don't know enough about her results, but she was able to detect differences in the breakdown of residue in the same market class.
so what she found was, so she did a similar thing that Taylor and I did, where, do you remember when we presented the soluble, the lignin, the hemicellulose, and the cellulose? Basically what you use that is you borrow procedures from forage, from the animal science folks that have a digestion to look at the quality of forage, but it also gives you a proxy for your soluble, your hemicellulose, cellulose, and lignin. But they group it together with these, it's NDF, ADF, I don't know if you're familiar with that in ADL, but Tammy found that decomposition rate was related to what we call ADF, which is primarily the amount of cellulose and lignin combined. And so that was a significant correlator.
But to get back to the question is, I think both varieties can accumulate nitrogen differently, I would assume. And it also has a lot to do with environment, which is mostly what Tammy had found in the different rainfall zones, that even the accumulation of these fiber components changed when the environment changes. Mm-hmm. So it's fairly variable.
So the question is to apply a product, a nitrogen product, yeah, to enhance breakdown. It's nitrogen limited, so that would help, because it's a nitrogen limited system, it's too carbon rich for it to break down. I'm not too sure about the proximity though, or in no-till systems having that stratification. I'm not too sure about that. Do you know?
There's just a lot of folklore out there.
So the question was, has anybody started looking at the effects on lower down in the soil from canola being grown in your rotation? Well, the answer is yes, we're definitely interested in that. But I touched on it a little bit through my thesis work, but we will definitely have to get farther into it.
Yeah, I don't know of anyone that's done aggregate stability. Bill, do you know anyone that's done any aggregate stability work? But aggregate stability definitely is an indicator of soil health. It's one of those physical, physiochemical qualities that would be routinely done, let's say if you were following the NRCS soil health protocols.
We actually have about 100 cores in storage. That's the intent. We're going to look at aggregate distribution, but also then look at the microbial communities associated with different aggregate sizes. Our approach was to see if communities change with aggregation, assisting aggregation, but you could maybe draw the same conclusions from that kind of work.